Experience built on thousands of replications of confined field trials with biotech events dating back to 1987 has allowed regulatory authorities in the United States to have high levels of confidence in the integrity and transportability of the data generated. These trials have been conducted without a single documented case of harm to the environment, to animals, or to humans. The testing program is extensive. Each year, the U.S. Department of Agriculture issues thousands of permits for biotechnology activities that includes importation and interstate movement of germplasm. For data generation, these permits allow confined field trials, including both commercial research trials and regulatory confined field trials. Each permit can include authorization for planting on a large number of acres, potentially at many different locations and geographies, and may include authorizations for multiple biotech events. Dr. Andrew Roberts is well-versed in these protocols, having spent the last 15 years working on biotechnology regulation and environmental risk assessment.
The Agriculture and Food Systems Institute, where I work, is a nonprofit scientific organization. And we have a large presence in the biotechnology space where we work with various stakeholders, including government, product developers, and researchers to help them better understand environmental risk assessment, and in particular, what data is useful and how it's collected and used for environmental risk assessment.
Roberts notes that regulatory confined field trials have a single purpose.
Regulatory confined field trials are conducted to support regulatory applications, and they do that by collecting data that's used for comparative assessment. Those comparative assessments address both particular characteristics of the plant that may be important for environmental risk, such as growth and reproductive habit, but also a variety of parameters that are measured and compared to non-genetically engineered varieties, just to ensure that there are no unintended effects related to the genetic engineering and breeding process.
This graphic shows an example of a confined field trial of a GM soybean. In this example, the confined field trial includes a GM test variety, a non-GM control variety, and 4 non-GM reference varieties. Each block is replicated 4 times across the trial, and plots are separated by buffer rows. Dr. Nick Storer with Corteva Agriscience has more than 20 years of experience navigating U.S. regulatory requirements for CFT and biotech events. He stresses trial focus is always on data integrity.
Data integrity involves knowing exactly what seeds were planted and which plants data are collected from, so that the data can be traced directly from the field to the final study report. We accomplish this by following good laboratory practices, or GLP. Part of GLP involves regular in-study audits by certified GLP auditors that are conducted to ensure that the data being collected are accurately reflected in the data being reported. These audits also are conducted on the final study report to ensure that the data being reported can be traced back to the individual plots, the individual plants where the data were collected from. In addition, all regulatory confined field trials are subject to inspection by the USDA to ensure compliance with the USDA permit conditions, including the integrity of the field trial site.
Storer says another factor contributing to the integrity of data is excellence through stewardship, a global organization with 60+ members providing guidelines for the entire life cycle of biotech products. Members' testing practices are audited, and the guidelines improve consistency of protocols and data being generated globally. The wide variety of testing conditions also bolster the global utility of U.S. data, according to Storer.
Regulatory studies are not intended to be conducted in environments that mimic every possible cultivation geography, but by including a broad range of different environmental conditions, regulators can be confident that the data being generated are applicable to their specific environment.
This graphic shows an example of confined field trial locations for a GM soybean. Trials are conducted across sites within the typical soybean production region. Sites are selected to represent many different growing conditions, such as different average temperatures, different amounts of precipitation, and different soil types. Roberts reinforces reasons for transportability of U.S. regulatory field trial data.
I have a high level of confidence in US-based regulatory confined field trials. First, the trials are being conducted by professional organizations that conduct a lot of these trials, understand the data that's being generated, and how the trials need to be conducted. The second reason is that the trials are conducted at multiple locations representing multiple geographies and differing environments, often over multiple years. Combined, this gives me a high level of confidence that the data being collected are of high quality and reliability.
USDA oversight, rigorous testing protocols, including adherence to excellence through stewardship guidelines, professional staff, data integrity, and a diverse range of testing environments, all bolster confidence in global transportability of data from U.S. trials.